Greetings, friends. This is our first podcast in which we are actually going to go into the details of the process of cell respiration itself. Cell respiration is we've had an introduction to it in terms of the basic idea of it and what's involved, and what you probably should have taken from that one mostly is the idea of oxidation reduction reactions, where we take electrons from one molecule, give them to another, and we use that energy that we transfer from one compound to another to do work. That's really foundational to cell respiration, or oxidation reduction reactions. The other podcast we've seen so far is about just the nature of ATP, and being as ATP is the whole point of cellular respiration, that's kind of important. So what we're going to do now is we're going to look exactly into this process of creating ATP. Okay? It is a complex process, and so there's some things that we need to keep at the foundation of that. And before we go anywhere, I want to make sure that we understand some really foundational concepts. First of all, this concept, biochemical pathways. We touched on this a little bit when we talked about enzymes back in the biochemistry unit, but we need to revisit it. A biochemical pathway is simply a long series of chemical reactions where you start with some substrate, and an enzyme helps you convert this substrate, I'll label it S, into some product that you need. But then perhaps another enzyme turns this product into a different thing that you need, and then maybe another enzyme, until you get to some ultimate product that you need. You end up with this long pathway, each step of it being catalyzed by some chemical reaction until you get to some desired end product. Okay? Cell respiration is a huge, long biochemical pathway. And if you take some sinister, evil cell biology course in college, you may have to memorize all the steps of it. We are going to memorize, or hopefully understand, important concepts within it. Okay? So, please, at the bottom of all of this, remember the point of all we are going to talk about is for the cell to make ATP. Okay? That's the point. Now, there is a let me give this overarching picture of where we're going, so you'll have at least this sketch in your mind. Now, first of all, we are talking about cells, but a very important part of a cell where cell respiration happens is mitochondria. Okay? So mitochondria is an organelles. Mitochondrion is singular, is plural. Mitochondria are where cell respiration occurs. Okay? Now, you need to understand about the structure of mitochondria. Mitochondria are double membrane organelles, and the inner membrane is highly, highly folded like this. Okay, so these are double membrane structures where the inner membrane is highly, highly folded. We are going to talk more about the structure of the mitochondria later when it becomes more relevant to us, but for now you need to understand that the bulk of cell respiration, CR, occurs there. So that's where the ATP is made. Now, having said that, the place where cell respiration starts is actually outside of the mitochondria in the cytoplasm of every cell. In the cytoplasm, requiring no special organelles or anything, in the cytoplasm is where we start cellular respiration. And we start it with this process called glycolysis. This diagram is really a good one. It shows you a mitochondrion here in this beige, yellow, whatever. It shows you glycolysis is the first part of this process, and it actually happens out in the cytosol, the cytoplasm. But this process creates products that get sent to the mitochondrion, and then the rest of the cell respiration process occurs in the mitochondrion. We'll talk about all this stuff later. Right now, our focus is on what happens in the cytoplasm, which is glycolysis. Okay? So, this slide is basically showing you, let me get all the words up here. There are two fundamental types of respiration. One of them requires oxygen, and that's aerobic respiration. Okay? Respiration using oxygen. Another type is called anaerobic, and you can do this without having any oxygen present. Glycolysis is anaerobic. Cells can do glycolysis without having to breathe oxygen. So that's really an important thing to remember. Everything else, aerobic respiration that we're going to learn about later, requires oxygen. Okay, now let's get on with what glycolysis is. Glycolysis, remember, foundationally, we are talking about breaking down glucose to get the energy out of glucose. Glucose is a great source of energy because it's got lots of carbon-hydrogen bonds, and those are high energy bonds. We strip the electrons out of all this, which means we remove hydrogen atoms, and we're going to get energy out of this molecule. So glycolysis is where we are going to begin to break down the glucose molecule. Glyco, sugar, lysis, split, break apart. We've heard lysis before. Okay? Now, this isn't going to have any meaning for you right now, or not much, but essentially what happens is this glucose, which is a sugar that has 6 carbons. Remember, every bend represents a carbon. Okay? And then there's a carbon up here, 1, 2, 3, 4, 5, and I'm missing a carbon somewhere. Where have I missed my carbon? I'm not sure. But at any rate, this thing has got ultimately 6 carbons. What we're going to do is we're going to break it in half, and we're going to get 2 halves, each half with 3 carbons. Okay? So we're going to snip this glucose molecule essentially in half, ultimately, and we'll see how that works. This is a really ancient pathway. Remember, I said this is the only part of cell respiration that does not need oxygen. So O2 is not required here. That means that this was evolved very, very early in the history of life when there was not a whole lot of oxygen present. There weren't a whole lot of fancy organelles evolved yet in cells. This part of cell respiration does not require fancy organelles, so it's a very, very ancient pathway. And the starting point for all of cell respiration. That's an important concept. Now, this is another important concept. It's not very efficient. We've got a whole lot of energy available in this glucose molecule, but we in glycolysis aren't going to retrieve a whole lot of that energy. So a lot of it goes to waste. That's important because the cell respiration process does improve on that later on. Essentially, from this, we are going to get 2 measly ATP for every single glucose that goes through it. That's not a whole lot. If you'll remember, 1 ATP is worth only 7.3 kilocalories per mole, and a glucose molecule, I'm going to have to go back and check my numbers, but I think is worth somewhere around 386 maybe kilocalories per mole, so we're not getting very much out of this process. Okay? And you do need to definitely remember this is occurring in the cytosol, in the cytoplasm. It's not happening in a fancy organelle. Again, it's suggesting a very early appearance, evolutionarily speaking. Okay? So, prokaryotes were the very first cells, if we can keep that in mind. Remember, prokaryotes, we're talking basically about bacteria. Bacteria are simple sacs of cytoplasm with DNA floating freely around, not encased in a nucleus or anything. There's no organelles. Also, when the first bacteria arose, there was no oxygen. Anaerobic atmosphere means there was no oxygen in the atmosphere. So no organelles, no oxygen. It makes total sense that glycolysis requires no oxygen and no organelles. And it makes sense that this is the first process for energy production that arose. Okay? Prokaryotes evolved glycolysis, and they are the ancestors of all modern life, which is why we still have this process. This is a direct hand-me-down from the first life forms. Okay. Please understand that every cell on Earth that we know of utilizes glycolysis. Okay? All right. Basic overview. I remember this term, biochemical pathway. Glycolysis has 10 steps in its pathway, 10 reactions, okay? With the ultimate goal being to take a 6-carbon glucose molecule and split it in half into 2 molecules called pyruvate, okay? And we are going to do this splitting in half of this molecule, not in one tidy step, but in several steps catalyzed by enzymes. Okay? Oddly, some of these steps are actually going to cost us ATP. We're going to have to invest ATP to do some of these things that we need to do to get to this point. This is only getting us part of the way there. We have to invest ATP to do that. Okay? Now, once we have invested our ATP and we have managed to split this molecule in half, which essentially we do right here at this step, and I'll show you some more about this in a minute. This G3P molecule, it's also called glyceraldehyde 3-phosphate down here. Okay, this is the one that's going to go into the rest of the process. But notice, since we split the molecule in half here, then everything that happens after this happens twice. Once to this half and then once to this half. This half will get converted to G3P and it will go through the rest of the process. So this will be a little bit more clear, but understand that once we split the molecule in half, then the rest of the process happens to each half of the molecule. Okay? Now, once we get it split in half, then it's a matter simply of converting this to pyruvate, which is the end product of glycolysis. We're going to use one of our electron carriers we talked about, NAD+, to help us with that. And we are going to end up, bottom line, generating 4 ATP. Now, if you'll recall, we spent 2 ATP up here. We're getting 4, so really all we're getting is 2 because we had to spend 2. Okay? So, overview. 10 steps, 10 chemical reactions with the goal of converting glucose to pyruvate at the end, producing a total by the end of 4 ATP, 2 NADH. We're going to pick up some electrons and convert an NAD plus molecule into NADH. It consumes 2 AT, so really all we gain from this process is 2 ATP and 2 NADH. And you might be saying, well, we want ATP, what good are these? Well, remember I told you if we follow the electrons, we follow energy. Something's going to happen with these electrons, and we're going to find out about what that is later. But for right now, we'll just accept it. Now, please note every step of this pathway is catalyzed by an enzyme. If you don't have an enzyme at any step in this pathway, the pathway dies. It's a goner. So, enzymes working in this pathway are absolutely critical for life to function. Okay? If you have something wrong with one of these enzymes, you're not going to do well. Alright, so glycolysis basically has 2 halves to these 10 steps, the first 5 and the second 5. The first 5 is an energy investment stage, meaning we're going to have to actually spend ATP to get this thing going. So far it's a really crummy process. If we're supposed to be getting energy and we have to actually spend it, put it in to the process. So initially it's actually endergonic. We're going to have to spend ATP to make it work. Okay? Now, once we get past that though, then we get to the exergonic part. This is the part where we actually get a little ATP to use for our cellular processes. Okay? And we also get NADH, which is going to be important, though you won't really understand why just yet. Okay? So here's our summary of our net yield. Okay, glucose, from that we're going to get some pyruvate, we're going to get 2 ATPs and 2 NADHs. Okay, and why, and well, let's just move on. I want you to see these steps and understand that there is no memorizing of steps. You may save that for real college, not pretend college that we do in this classroom. Okay? The first half of glycolysis is simply getting the glucose ready to split and then splitting it. Okay? So, let me show you what happens. Glucose enters in to the process. We're going to spend some ATP, and I don't know if you remember what we said about ATP, but ATP, when we have those adenosine with its 3 phosphates, and we strip one off and stick it on another molecule, like you can see has been done right here, this phosphate was taken off and stuck to glucose, and now glucose looks like that. What this does is this phosphate destabilizes that molecule. It's going to make it a little bit easier to break it. That's why it's worth it to us to spend an ATP there. Notice here's an enzyme, hexokinase. You don't need to remember it. I'm just showing you that an enzyme has to be present to catalyze this step. Another enzyme here changes this into something else. Fructose 6-phosphate, don't care, is created. So this is the original substrate. This is a product. But notice that this product becomes a substrate for the next reaction. And this product that we create becomes a substrate for the next reaction, and so on. This is a typical biochemical pathway. Now, notice here we're going to take some more ATP, we're going to take a phosphate off, and we're going to stick it on. So now not only do we have a phosphate over here on the glucose, which is now not glucose anymore, but it's fructose, and we're going to stick a phosphate here. Now this thing's getting really unstable. It's got all these phosphate groups on it. And what's going to happen is we're going to break it in half. Okay? We're going to break it in half. And what you need to understand is this enzyme can basically take this and turn it into that, or take this and turn it into that. Because G3P is going to go on into the rest of the process, it's always going to be pulled out and fed into another process, so equilibrium is going to cause this to get pulled over and changed into G3P. We almost never get it going back this way. This is a minor point, but this is why the arrow's going to keep going and taking things in this direction. So, bottom line, first step, we have split a destabilized glucose. It got destabilized because we added phosphate groups from ATP molecules to it, and it broke in half. And what's going to happen is G3P, two G3P molecules ultimately are each going to go through a second set of chemical reactions. So from this point forward, we need to multiply everything times 2, because 2 halves are going to go through this entire process from this point forward. Okay? So here is the second half of glycolysis. Now this is where we're finally going to get some bang for our buck. 5 more reactions, again, each catalyzed by an enzyme. G3P is coming in from the other side. Please notice that in addition to this enzyme that's going to have to catalyze this reaction, we also need, in order to make this go, our electron carrier is going to have to come in and carry some electrons, pick them up and carry them away in the form of NADH. We're going to have ADP come in, ADP come in, pick up a phosphate, and it's going to become ATP. Okay? So, this is actually showing you both G3Ps coming and doing the same thing, so you can actually see it happening twice. Most diagrams don't do that. Usually most diagrams just show you one thing and tell you to multiply it by 2. So be aware of this diagram is more literal than most. So, what is happening here is that we are actually going to get an ATP here. Okay, we're going to take that phosphate, one of the phosphates, and stick it onto an ADP molecule. More enzymes are going to change this thing, the remnants of this glucose molecule, some more, until finally we get to our last step, in which this molecule gets converted by pyruvate kinase. Remember, a kinase is something that phosphorylates something else. Well, this is going to again take a phosphate from the substrate molecule we have up here and stick it onto ADP, so we get ATP. So, bottom line is this. We're going to make some NADH. This is important because it's got electrons on it, and remember, electrons mean we've got energy. It's going to do something for us, so that's important. Okay? We have stripped electrons from the sugar molecule. We've oxidized sugar. We've taken some of its energy. We have reduced NAD+. We gave it electrons, turned it into NADH. Okay? We've also made some ATP. By turning G3P into pyruvate, we have taken phosphates and stuck them onto ADP molecules. Okay? Now, I want you to know this phrase, substrate level phosphorylation. An enzyme, a kinase, physically strips a phosphate from somewhere, okay, from this molecule up here, and it sticks it onto another one, in this case the ADP, and turns it into ATP. So this will be more contrasted with a different type of phosphorylation we're going to do later. But in this case, physically, we're going to strip a phosphate from a substrate, stick it onto an ADP, and bingo, we've got ATP. Okay? And that happens in 2 places. It happens up here, and it happens down here. Okay? So, finally some ATP, but heavens to Betsy, do you see how much work that was to go through all of this? Remember, we had the previous page as well. All of those reactions just to get, in the end, 4 ATP. But remember, we had to spend 2 in the first half. So really, we got to subtract the 2 we spent, and all we're getting is 2 measly ATP for all this work. It's madness. It's madness. So there's got to be more ways to get ATP from glucose than this, and there are. Okay? All right. This is going into some detail on this idea of substrate level phosphorylation. Basically, this is really showing you detail of how that can happen. Here's the enzyme in purple. This enzyme comes in, holds an ADP molecule, holds that phosphenyl pyruvate, which you do not need to know, but they're calling it a PEP molecule. And if you'll notice, the PEP molecule has a phosphate group stuck onto it that was put there earlier. Well, this enzyme causes this phosphate to come off and stick onto here, to ADP, and thus, once that happens, we have, ta-da, ATP. And we also have a pyruvate molecule. Okay? So, those are our creations from substrate level phosphorylation. A enzyme rips a phosphate off a substrate, in this case the PEP molecule that's right at near the bottom of the glycolysis pathway, and sticks it on to ADP, and we get ATP. Okay? This enzyme, therefore, would be a kinase, something that phosphorylates something, sticks a phosphate onto something. Okay? So the phosphate came directly from a substrate. Alright, this is just more going through where what you get out of glycolysis. We've already said it many times. It is important. They do like to ask questions about this. What's really important is that you understand how little energy it is. But we've spent 2, we get 4, but because we spent 2, we are really only going to get 2 ATPs, plus these very important little taxicabs carrying their little electrons, these little NADH molecules, these electron carriers. They are carrying those electrons somewhere, by golly, and we'll find out where soon. Okay? So those are your net gains, your net investment. So basically now the bottom line is we got our little bit of energy, and we also converted a 6-carbon sugar, glucose, into 2 smaller 3-carbon sugars and stole a little bit of their energy in the process. So indeed, all that work, and that's all we get. But glucose does have a lot more to give. Okay? But for 1 billion years on planet Earth or more, this is what life did. This is all they had it. And if you're a prokaryote, it's okay. It's all you need. There are many prokaryotes that still use this as their only means of obtaining energy. Not until we got a lot of energy in the atmosphere could natural selection begin to favor a way to get more energy. So oxygen, the deposition of oxygen in the atmosphere by plants doing photosynthesis really was very, very important for the advent of cellular respiration. Okay? Let's see, I'm trying to decide if this is anything we need. You know what? We're going to not do this right now. I'm skipping over this. Skip, 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 skip. We don't want any of this. We're going to talk about fermentation later. Skipping, 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 skipping. Going to talk about that later. I'm just trying to see if there's anything else I need to tell you about. All right, I don't think so. Next coming up for us is going to be learning the story of this guy, the mitochondrion. Remember, everything we just saw took place in the cytoplasm. Everything we see from here on out relating to cell respiration is going to be happening in the mitochondrion. So that's our next stop, and where we're really going to start seeing, I hope, lots of ATP. All right, thanks for watching, and I'll see you in the next podcast.